Good afternoon, students. Hope you all are fine. As a part of your core paper corresponding to microbial genetics, till now we all have studied about mutation, about various subtypes of mutation occurring at the translational level, like silent mutation, nonsense mutation, missense mutation, and about the techniques meant for selective isolation and screening of mutant colonies, like replica plating technique, crowded plating technique, penicillin selection method or negative selection method, etc. Now let us move on to the next part of our study, or next topic of our discussion. I'll divide my today's lecture into two segments. In the first part of my lecture, in the first segment, I'll be going through how mutation actually differs from recombination and how the two processes fuel or carry out the process of microbial evolution. I'll put focus over few more special forms of mutation other than missense, nonsense and silent mutation. And for the second part of my lecture, I'll be putting a huge focus over some phenomena involved in mutation, actually causing mutation, or in brief, I'll be going through the molecular basis of mutation. So we all know that mutation is any heritable change in the genetic constitution of an individual. That is, by mutation, we mean to refer to a change in the nucleotide base sequence of the genomic DNA of an organism. Mutation brings about or leads to a very small change. It's not in magnitude, its effect is very small. The size of change may stay restricted to one or two nucleotide base pairs of a genome. And mutation brings about changes, some of which are good, while others are bad. Now, is there any other way by which such a change in the genome can be brought about? Yes, there is. The other method which happens to occur in that, or by which we can bring about a change in the genome of an organism, is recombination. Other than mutation, recombination also brings about such a change and fuels the process of microbial evolution and generates variation in a genome. So what is recombination? Recombination actually involves two different organisms and is a method of horizontal gene transfer or lateral gene transfer, in which then occurs a share of genetic material between two organisms. One, called the donor, donates a piece of genetic material which is called exogenous or foreign DNA, and that exogenous DNA in course is then received by the recipient cell. Hence, recombination is actually a process which involves two different organisms as a process of genetic exchange involving lateral gene transfer. And what is the effect of recombination? The effect of recombination is that entire set of genes or even genomes are transferred from one organism to the other. So recombination, while mutation produces a very small change, recombination leads to a huge genetic variation in the genome of an organism, and is very large in size and magnitude with respect to its effect. So, the similarity in mutation and recombination is that these are the only two methods available which can generate variation in genome and which thereby fuel the process of microbial evolution. So today, I have decided that I'll make you all very clear that how does mutation differ from recombination. The similarity is that both are the methods meant for generating variation meant for generating variation in genome of organism. But therein lies a huge line of difference, and therein lies a sharp contrast by which recombination differs from the method of mutation, or mutation differs from recombination. Number one is that mutation involves a single organism and is the change in the genome of an organism, whereas recombination involves two different organisms. One is the donor, which donates genetic material, a piece of DNA, exogenous DNA, and the other is the recipient, which receives this exogenous DNA. The second major difference is that the effect of mutation is very small. It stays restricted to a single nucleotide of a genome, while the effect of mutation is very large. Recombination is very large. Recombination is actually may be defined alternatively as a process in which the genes or the genomes from two different organisms are brought together in a single molecule. So you can imagine that what is actually the size of the genetic material that is getting transferred during recombination. At the end of this process, entire genes, set of genes, or even genomes are transferred from one organism to another organism. Hence, unlike mutation, recombination involves a very huge and a large chain variation or change in the genome of an organism. And the third thing is that mutation occurs spontaneously in nature, mostly occurring in all living cells, or is brought about or induced by certain agents called mutagens. But how does recombination occur? Recombination occurs as a result of gene transfer from the donor to the recipient that is being mediated by one of the three methods, or occurs in the form of one of the three methods, either transformation or transduction or conjugation in course of the horizontal gene transfer or lateral gene transfer. What is it? Horizontal gene transfer is the transfer of genetic material from donor to recipient. And how does it differ from vertical gene transfer? Unlike vertical gene transfer, it does not involve the transfer of genetic material from parent progeny to daughter cells. So vertical gene transfer specially refers or exclusively refers to the transfer of genetic material from parent to daughter cells. So these are the major differences between mutation and recombination, and this is how a mutation differs from recombination with respect to the organism involved, the way or the molecular basis, then with respect to its magnitude or size and the effect it generates, etc. We have already gone through various special subtypes of mutation occurring at the level of protein translation, like missense, nonsense, silent mutation. Today, I'll take you through few more various subtypes of mutation, which are also very special. You might have heard about conditional mutation, isn't it? Have you? Yeah. By conditional mutation, we mean to refer to some kind of mutations which are only expressed under certain environmental conditions. Supposedly, say, an E. coli cell is said to be conditionally lethal under high temperatures only when it grows under permissive or cold temperatures, but expresses the effect of mutation, and as a result of the effect of mutation dies when exposed to restrictive temperatures or high temperatures. So this is an example, or such a mutation is conditional mutation. Biologist John Keynes and his group of collaborators, while working with mutation, came out with a very interesting subtype of mutation, which is called directed mutation. It's also called adaptive mutation, and the cell in course of directed mutation prefers to choose and go for a certain category of mutations which would help the cell to adapt and to survive better in the surroundings and environment. Supposedly, say, an E. coli cell which experiences a directed or adaptive mutation, all of a sudden is able to utilize a carbon source to which it had experienced a kind of intolerance previously. So supposedly lactose, by exposing an E. coli cell to lactose, lactose is made tolerant to that E. coli, and the cell can now grow and proliferate utilizing lactose as the only or the prime source of carbon and energy. So this is a very interesting example of directed or adaptive mutation. Now, one more form of mutation, which is very subtype of mutation, it's very interesting and very common, is hypermutation, when a group of mutations assemble and together and generate their effect. And this hypermutation is mostly known to occur in response to starving conditions. When bacteria during starving conditions are devoid of nutrients, they experience or show the effect of a kind of mutations and show a variety of mutations which occur as a result of activation of a group of mutated genes. Several such mutated genes have been very well studied, causing hypermutation in bacteria and in, of course, other organisms. So, we came across, we saw that how mutation differs from recombination. We saw on various subtypes. We studied about various subtypes of mutation, like conditional mutation, directed adaptive mutation, hypermutation. So now that's all for this part. I would ask you all to study more about all these topics from two books. The first one which I'd be referring to is, which I'd be asking you to go through is Broad Biology of Microorganisms. And the second book, you can take the help of one more book, that is General Microbiology, that's by Prescott, Harley and Tin. So see you all in the next part of the talk, next session of today's talk. It would be all about molecular basis of mutation. Till then, keep studying and stay tuned. Goodbye.